Hello, sir.
Hi, how are you?
Good, sir.
Looks like you want to give an update, or?
Yeah. So basically, I was looking into a few things. Like I was talking, 2 weeks earlier, probably. So I was reading a couple of papers for, this biological inspired neural networks. I've been seeing them, and I have recently seen, this update for the GSOC, and there are like 2 projects that have been opened up in the space. So I wanted to, focus on the 2 things. The first is I wanted to see what can I contribute to the project, and parallelly read about the biologically inspired neural network. So anyway, the motivation to do like the biologically inspired neural networks, that anyway keeps going. So research is a long run. So we have so much time for that. So I wanted to do that parallelly. But so in this timeline, I wanted to focus on the project part. What can new things can be added into that space. So I wanted to look into that too. So that could be like my initial side of things, and probably in like a 2 or 3 days, I have read the paper that was published. So in the archive, you have given me, sir, that's the biologically inspired network. So it was 2017 paper, like ANNs and BNNs, like biological neural networks. So I have seen certain literature from neurocomputation, that paper, and I have also seen certain literature from like other journals and conferences. I wanted to add up into that paper, like few things got emerged into like the recent line of neural networks, like hypergraphs and all. And there are like certain unsupervised or self-supervised methods which actually do not need labels to learn certain features. There are so I wanted to extend the line of work in that that paper. So I've seen certain gaps that we can address and then improve that work on. So I've seen some space where we can add some new things into that. So I'll probably share with you in a couple of days, like what we can do in that. Yes, sir.
Good. Yeah. All right. Thanks for the update. Okay. Does anyone else have any updates they'd like to share? So let's get started. All right. So first of all, I wanted to give a congratulations to 2 people who have been here in the group, or haven't been around for a couple of years, but so this is Mayukh and Mainak Deb. They're brothers, and Mayukh is in the School of Psychology at Georgia Tech, Cognition and Brain Science, and Mainak, I guess, is still an independent contributor, and they published this archive paper on TopoNets, and it's TopoNets, high-performing vision and language models with brain-like topography. So Mayukh has been pretty active in this space of, machine learning and thinking deeply about like machine learning and deep learning. And, he's, I think, somewhat interested in biological neural networks, being in a brain and cognition and brain sciences program. But he's done some pretty interesting writing on this. So this is this article. Let me zoom in. So I'll read the abstract. Neurons in the brain are organized such that nearby cells tend to share similar functions. AI models lack this organization, and past efforts to induce topography have often led to trade-offs between topography and task performance. In this work, we present topo loss, a new loss function that promotes spatially organized topographic representations in AI models without significantly sacrificing task performance. Topo loss is highly adaptable and can be seamlessly integrated into the training of leading model architectures. We validate our method on both vision, it's ResNet 18, ResNet 50 VIT, And language models, GPT-Neo 125M, Nano GPT. So they basically have this method, they validate it in these different models, and they call it TopoNets. TopoNets are the highest performing supervised topographic models to date, exhibiting brain-like properties such as localized feature processing, lower dimensionality, and increased efficiency. TopoNets also predict responses in the brain and replicate the key topographic signatures observed in the brain's visual language cortices. Together, this work establishes a robust and generalizable framework for integrating topography into leading model architectures, advancing the development of high-performing models that will more closely emulate the computational strategies of the human brain. So that's his, abstract, and they're at this, I guess this is the Murty lab, this is Murty, the person running the lab here, as the third author. So this is very much this sort of comparison between biological and artificial neural networks. Neurons in the brain are not tossed around haphazardly, which is, what they mean by that is that they're spatially organized. If you look at a micrograph of the brain, it looks somewhat haphazard, at least at first appearance, but there is a spatial organization that occurs in the brain. Just like in the embryo, you have this spatial organization. They're spatially organized such that nearby cells perform similar functions. Topographic organization is a core feature of brains, meaning that you get different regions that do different things, or you have different cell types in different regions. You don't find any one cell in any other region, that sort of thing. In visual cortex, this organization is evident in microscale pinwheel patterns for orientation selectivity. So this is in visual cortex, where you have these pinwheel patterns that organize for, allowing an organism to orient to different places visually, and they have this tuning function. And orientation selectivity occurs at macro scale, allows for macro scale category selective regions for faces, bodies, scenes, etc., and then large scale organizational biases for real world shape, size, curvature, and animacy, which means movement. So there are these sort of representations, or I don't want to say representations as much as maybe spatial structures or spatial orientations of collective behaviors of cells that work towards this sort of functional aspect. And the functional aspect, of course, is for different things like different objects. And so you should expect a certain response from the network for these different functions. So for face perception, for body perception, for scene perception, etc. And then these different physical features in the image. Beyond vision in the language cortex, recent studies have also identified clustering of neurons with distinct temporal integration windows. Unlike the brain, most artificial neural networks, or these ANN models, lack any kind of systematic organization of units. So this is where, these artificial neural networks are sort of periodic, but also random at the same time. So you don't have very much structure. You don't have this sort of, these regions or these structures that yield some sort of function based on their topography, basically. So this is what they're kind of focusing on here. In this work, we introduced topo loss, a new brain-inspired inductive bias that can be easily integrated into the training of most current AI architectures, or ANN architectures, both convolutional and transformers. The resulting model TopoNets exhibit brain-like topography characterized by localized, low-dimensional, and efficient representations. They'll show that in Figure 1. Inducing topography into artificial neural networks has been proven to be challenging. Two major strategies have emerged. So this is in general trying to, create these sort of heterogeneous ANNs. So the first is post-hoc topography, where you have reorganizing units that fit into these pre-trained models using methods like self-organized maps. So you have these self-organized maps that play a role in guiding learning, especially pre-training. So self-organizing maps are kind of an older tool from Kohonen and then developed later on. And they're basically these unsupervised learning methods where you have an array of cells and they kind of take on this topography based on what they've been exposed to or what they're learning. So you actually have this pattern that emerges from the specific training regimen or training data set that it's exposed to. So that's post hoc topography, basically where you don't define a topography in advance and you let it sort of emerge from the interactions. The resulting models exhibit topographic signatures, but the underlying representations remain unchanged from the original model. So you have this sort of basic goal that you want, this representation, and you try to match it and get these topographic structures. Consequently, the functional advantages of topography, such as reduced dimensionality and increased efficiency, are not realized. The second strategy, jointly optimized topography, incorporates an additional topographic loss during model training. These models induce topography by either 1, explicitly matching the brain's spatial correlation structure, 2, imposing distance-dependent constraints, or 3, encouraging information redundancy. So these are this is the jointly optimized topography approach. These approaches suffer a significant trade-off. So they're better, I guess, than the self-organized maps, and they're better than sort of a homogeneous random network. But there is this trade-off. So the trade-off is the ability of the model to learn task-relevant representations. So sometimes it's compromised, and sometimes, you get really good performance, but you also get this this task relevancy. So they perform poorly on engineering metrics, like performance on ImageNet, for example, and or show diminished capacity to predict brain data. Most prior work in the space has been focused exclusively on vision models, and the one attempt at imparting topography to language models focused on self-attention maps of BERT, which is an AI model. It's a language model, which resulted in only modest topographic organization in the output space. So this is where you have this trade-off between sort of performance and specificity, and it's hard to kind of get a balance between them. To summarize, there is currently no unified strategy to apply topography across common ANN architectures, such as convolutional nets and transformers, and domains, vision and language. So you have these different sort of architectures, you have these different domains of learning, and there's no strategy to apply these across these common architectures and domains that can deliver high-performing models together with the functional benefits of topography. So we're looking for the performance gains that we might get, but also this functional benefit. So, why use a topographic approach if you can just use a sort of a random approach better? What's the benefit of it? Okay, so the figure 1 is here. This is basically talking about TopoNets. This shows transformation from unstructured baseline models on the left to organized topographic representations on the right. So you have these unstructured models on the left, which are the baseline models. You have topographic vision models on the top, such as ConvNets and transformers. This includes ResNet 18S, ResNet 50S, VITs. So there are different types of transformers, different types of neural nets, convolutional neural nets. And this just shows that there's no structure to it. It's just that it's learning. And then you have TopoNets on the right, which are models trained with topo loss, brain-like topographic organization. So you have these regions that are devoted to specific things, and they're connected together like you would find in a brain. So that's the difference there. As it's learning, it's utilizing the structure to improve learning or to constrain learning or hopefully to allow for generalized learning. And so, I mean, the goal would be to make it look kind of like a mammalian brain, basically, or any kind of brain where you have regions, which are most brains. You have regions, language centers or vision centers or things like that, and the information gets passed between these centers, basically. And then on the bottom, we have topographic language models, which are transformers, GPT-Neo 125M, and Nano GPTs. On the left, again, we have the unstructured models, which are these where you have no organization, but you have learning. And then on the right, you have topographically organized units, which have this, again, the structure. And again, you want to achieve, task and variant learning. And you want to have something, hopefully, that integrates a lot of the multimodal information. So ideally, you'd like to have a structure that would allow for both visual and language learning instead of having a vision model and a language model. So that's figure 1. And looking through the rest of the paper, they kind of go through and define their cortical sheet. So their first task was to define a two-dimensional sheet of cells where we could apply our topographic loss or topo loss algorithm. To demonstrate that topo loss generalizes across different domains, we applied it to both language and vision models. For language, we trained GPT-Neo 125M on the Wikipedia dataset and Nano GPT models on 10 billion tokens of the FineWeb EDU. So this is where we have Wikipedia dataset and FineWeb EDU. Those are the datasets, and then the models are GPT-Neo and Nano GPT. For vision, we trained topographic Resonate 18 to allow comparisons with previous topographic models and Resonate 50 and VITB32 models to further evaluate generalization of topo loss to larger models and architectures. All vision models were trained on a supervised 1,000-way classification task on ImageNet. Together, these languages and vision models allow us to robustly evaluate topo loss. So that's they're defining their cortical sheet. Then they define the cortical sheet in the different models. So in the language models and VITs, you have this linear layer with I input units and no output units. We reshape its weight matrix to a cortical sheet. So this is from W to C, and this includes from output times input, so the interactions there, to H times W times D. So H times W times D, H times W is the area of the sheet, so it's the height and the width. That corresponds to the number of output units, corresponds to the number of output units, so it has this two-dimensional shape. And then depth D corresponds to the number of input units, or I. So basically, you take O and I and you transform it into this two-dimensional sheet with a depth. To maximize the number of neighbors for each element in the cortical sheet, we chose H and W to be as close to each other as possible, so you want a square sheet or close to a square, thereby minimizing the perimeter. Each element in the cortical sheet now represents the weights associated with a single output unit or neuron in the original linear layer. So they're making this transformation from the weight matrix, which has the weights for each sort of, this random sort of stationary matrix, to this structured non-stationary matrix in this way, this sort of geometric way. And you can imagine you can probably do this in different ways. This is not the only way to do it. In convolutional models, or what we basically think of as vision models, for a convolutional layer with C input channels and C output channels. So again, you have the C parameter, which is input and output channels, and a kernel size of k by k. We project its weight tensor, and a cortical sheet. So the weight tensor is W, the cortical sheet is C, where the area corresponds to the number of output channels, H times W, and the depth is defined as D, which is equal to the number of input channels and times the kernel size, basically. And in previous work, we arranged the model units, conventional kernels on a two-dimensional cortical sheet. More detailed explanation is provided later. So that's, and then inducing topography is where they do the topo loss. So the second step introduces topo loss to the reshaped cortical sheet, promoting topographic organization. We achieve this by maximizing the cosine similarity between the original cortical sheet and its blurred version. So they blur the original cortical sheet, and then I guess they're going to recover it and look at the loss. This suppresses the high frequency noise, leaving behind only the most important and meaningful information. This idea was motivated by synaptic pruning in the brain, which eliminates noisy high frequency neural connections, refining the biological network structure. Although note, we do not explicitly remove any weights here. So they have this this smoothing process where they get rid of high frequency noise. The blurring of a 2D signal can be defined using a downsampling function and upsampling function as follows. So they have this blur equation, and so they have the downsampling factors along height and width dimensions to encourage smoothness in the cortical sheet. We maximize the cosine similarity between C and its blurred vision C prime across cortical sheet layer maps. This process smoothens the representations and encourages topographic organization. The topo loss is defined this way, L topo in this equation here. This topo loss is integrated with the original training loss as follows. So it's the training plus tau topo, which is the total. Tau is a scaling factor that controls the strength of the topographic effect. Higher values encourage stronger topographic organization in the model. So again, we're taking topo loss, we're integrating with the original training loss of the original model, and we're doing it that way. So, they have this training for vision and for language models, and they specify it here. They also have effective dimensionality, which is this lambda I indicates eigenvalues and the number of eigenvalues. So you have a number of eigenvalues that you're looking at for the dimensions. This metric measures the spread of the eigenspectrum. For ResNets, we follow the procedure outlined in this reference here. For ResNets, we chose 20,000 images from the ImageNet validation set, calculated the effective dimensionality for all the convolutional layers. And then for language models, they did something appropriate for that. They smoothed it, they did this unstructured pruning, this downsampling. They estimated the selectivity, which is where you have these layer-wise features in response to stimuli, and calculating it using a standard method for these representations. And so then the results are TopoNets achieve high model performance with comparable spatial topography. And here is a graph here, figure 2. TopoNets achieve higher model performance with comparable topography. So let me so this is figure 2. Okay. So this is figure 2. This is where we have on the x-axis for both graphs, we have the degree of topography. We go from no topography to high topography. So smoothness decreases over space, I guess. Or I guess now smoothness increases because you're smoothing out the topography. And then ImageNet validation accuracy on the y-axis. basically you're validating your ImageNet data set. Higher is better, so the higher we go on the graph, the better we get. So we have these two results for vision and language models, vision on the left, language on the right. We see that we have these previous best topographic models on the bottom and in pink or in fuchsia, and then blue baseline models on the far left, which includes ResNet 18, VITB32, and ResNet 50. So you can see for these different tau values, they plot them out on these functions. You have these two functions that kind of go out, and so you have TopoNet ResNet 18, TopoNet VITB32, and then TopoNet ResNet 50 at the far right. And so those are the results there. And then for language models, we have the same thing with baseline models. We have TopoNet GPT Neo 125M, and then TopoNet Nano GPTs. So you can see that there's basically this trend towards high topography. And let's see if we can figure out more about this from the legend. So the dashed black lines indicate the Pareto curves for ResNet 18 and ResNet 50 models. And so the Pareto curves are these dotted lines. The y-axis here denotes the language model evaluation score in BLIMP. The dashed gray line indicates the reported topography from our prior study. Okay. So figure 3 is topography explains reductions in model dimensionality. So this is where they have vision models and language models, the vision models on top, and then the language models down here. So the vision models have this graph of model performance, which is ImageNet validation accuracy, the percent correct at the left, and then the degree of topography or smoothness on the right. And this is both in terms of this effective dimensionality or the eigenvalues. So for vision, you have these models over here. Model performance increases. You've ResNet 50 on the far right, ResNet 18 a little bit less accurate, VITB32 a little bit less accurate than that. And then you have these tau values plotted out at different points. On the right, we have these tau values. We have, so that's model performance. We have the percent correct degree of topography. Your VITB32, ResNet 18, and ResNet 50 are all kind of on the far left with a low degree of smoothness, and that's sort of a higher degree of randomness. So this is topography, not model performance, explains reductions in dimensionality. So the effective dimensionality is high for VITB32 and lower for the ResNet models. In the case of language, we have the same sort of thing. We have model performance in terms of BLIMP accuracy, and then degree of topography or smoothness on the right. So again, Neo 125M is very low model performance. Nano GPT, very high model performance. Nano GPT has a higher effective dimensionality than the Neo 125. And then for 4, measuring the efficiency of TopoNets against baseline models, you have these vision, and language example. You have ResNet 18, ResNet 50, and GPT Neo 125M. You have these fraction masked weights, so this unstructured pruning versus drop in ImageNet accuracy from baseline, and then TopoNets on the in B on the very right. That's where we have these language models, and so basically on the left we have the vision and language models, and their point A in this figure is that TopoNet show greater resilience to L1 unstructured pruning, exhibiting sparser weights, and in this B example, TopoNets are more parameter efficient, showing the example for downsampling here, where you're downsampling the topo layers. So yeah, that's I'm not going to go into any more data there, but I think you get the point that they did a lot of work, that they developed this technique for looking at these structured networks, and that they can test it for both vision and language models, and they get pretty good results with respect to their mathematical model and their baseline. So one more thing here. There is a actually this isn't. Oh, yes it is. Okay, so one more thing here. There's a GitHub repository, TopoNets high-performing vision and language models with brain-like topography. I don't have the GitHub repository up as an active link, but if you go to their GitHub repository, search for TopoNets, you'll find it. This was an ICLR spotlight for 2025. This is where they have the code, they have the implementation in PyTorch, they have some of the graphs here, and yeah, so check that repository out. So I don't know if we had any questions or comments on that. I know Lalith probably has something to say about that.
So why are we using GNN in this work project?
What was that?
I was asking that we had a project for using GNN this year for GSOC. So can you explain that in more details?
Why are we
What?
using graph neural networks?
Yeah.
Oh, well, in our group, we are focused on graph representations of developmental biology. So we're interested in the graph structure of different embryos or different processes. And moving from that, you can use graph neural networks to analyze data that have this graph structure. So that's where we've been using this before. We've also been looking at this issue of topology in development, because developmental structures have a high degree of geometric complexity. And so graph neural networks are interested in sort of how to analyze that. So yeah, I don't know how else to we have a number of different interests in the area. We haven't really published anything specific to implementing graph neural networks. We have the hypergraphs paper, the hypergraphs preprint, which is on the archive, and I'm going to share that with you in the Slack. I think it's already been shared in the Slack, but and we're preparing another version of this from last year's work, where we did C. elegans analysis of the C. elegans embryo using looking at hypergraphs and graph neural networks.
So could you please share the research paper link of the TopoNet that you just read out?
Yeah.
Explain us.
Yeah, I can.
Yeah, thank you.
Did we have any other comments or questions on that paper, on the TopoNets paper?
Sir, I have a small doubt regarding not the paper, but the paper is really interesting. So do we have, are these, the project that was, assigned or like whatever that's posted? So do we really need to stick with that? No, or we can, add some new things into that space? So do we have the bound for that, or are we open to do something new which can add up to that?
Oh, you mean like in the GSOC project?
Yes, sir.
Oh, well, yeah, the way I approach that is usually you can sort of come up with your own, like you can work within the bounds of the project description, and you can do kind of whatever you want if you think it's relevant to the overall goal. So, I don't know what you're thinking in terms of, how to contribute to it. I mean, contribution is just making it useful or making it better. So I don't
Yeah.
care if it's like involving different methods and how to get there. Because, we had different approaches to it, and they kind of coexist. So, yeah, I mean, think about like maybe how would I kind of work in the GNN space, but like how would I make this useful to myself? How could I make this useful to those of the group? And
Yeah.
you propose any sort of project you want. So I'm pretty flexible.
Sure. Thanks.
All right, so I don't know if we have any other comments or questions. So if not, well, let's move on. So this is an interesting paper. We've had some discussions on synchrotron X-ray tomography in the past and doing things with embryos. We did some things, I think, with axolotls and talking about different types of approaches to this, where a synchrotron is a large source of electrons, and they usually have them at some sort of super collider facility. And the idea is that you get these electrons are highly charged, and they allow you to image things at a very high resolution. And so that's why they're attractive to microscopists. So this is a microscopy oriented paper. This is from PNAS. This is not imaging embryos, but molluscan brains. So molluscans are, of course, marine vertebrates. And they're doing this functional mapping of the molluscan brain. And this is guided by synchrotron X-ray tomography. So I'm not familiar with this group, but let's go through the significance and the abstract, and then look at what they're kind of maybe, I don't know if they have results down further in the paper. So the significance here is, thanks to their accessible nervous systems, simple model organisms have provided some of the most fundamental insights into how neural circuits generate and control behavior. However, in many cases, understanding is constrained by the absence of detailed brain maps. Here we address this challenge using a synchrotron source to enable rapid high-resolution X-ray imaging of whole multi-millimeter scale brains. Applying this classic molluscan model, Lymnaea, we construct a detailed 3D atlas of its feeding circuit and use this to guide identification and functional characterization of pivotal cell types in the network. Our approach readily generalizes to CNS atlas building and other model organisms, enabling the interrogation and sharing of brain structures across research groups or comparative and functional studies. So what they're doing is they're constructing this atlas from these high-resolution X-ray images. They're looking at this molluscan model, which is fairly well characterized. They build this 3D atlas of a feeding circuit, so it's not the whole, they get information for the structure of the brain, but they're reconstructing this feeding circuit and then putting it into a detailed 3D atlas. And then, so then their idea is that their hope is that you can use this as a methodology for other types of neural structures. So let's see, the abstract. Molluscan brains are composed of morphologically consistent and functionally interrogable neurons, offering rich opportunities for understanding how neural circuits drive behavior. Nonetheless, detailed component-level CNS maps are often lacking, total neuron numbers are unknown, and organizational principles remain poorly defined, limiting a full and systematic characterization of circuit operation. So, this is we just talked about how you can use topographic structure in or you can use that principle in artificial neural networks to give you maybe a boost in performance, or to give you something that you're missing in these kind of stationary random networks. And what they're saying here is that we don't really understand that well in biological neural networks. We have to sort of kind of do a lot of imaging and figure out kind of from the basic structure what the information processing value of that structure is. So this is where we're trying to get at this. Here we establish an accessible, generalizable approach, harnessing synchrotron X-ray tomography to rapidly determine the three-dimensional structure of the multi-millimeter scale CNS of Lymnaea. Focusing on the feeding ganglia, we generate a full neuron-level reconstruction, revealing key design principles and revising cell count estimates upward threefold. So with this high-resolution imaging, they're not only able to get more information about the structure, they're able to get a re-estimate of the cell counts that in the brain. So they've been able to revise their cell count estimate upward threefold. So that's interesting. Our atlas uncovers a superficial but also non-superficial ganglionic architecture, reveals the cell organization in normally hidden regions. So they have these, when you have something in microscopy, you usually acquire a stack of images. You go through focal planes. Sometimes things are occluded in those images because you can only get to the things that you can, basically access through some sort of visual signal. So sometimes we use fluorescence. Sometimes we use different lighting techniques. And so they have these ganglionic dark sides, as they say, which are these hidden regions or occluded regions, which are now visible from the synchrotron approach. These detailed features of single neuron morphology, together guiding targeted follow-up functional investigation based on intercellular recordings. So they were able to reconstruct the circuit using this sort of visual approach, and then they were able to confirm these cells and their cell types using intracellular recordings of electrical activity. Using this approach, we identified three pivotal neuron classes. The first is a command-like food signaling cell type. The second is a feeding central pattern generator interneuron. And three is a unique behavior-specific motor neuron. These together significantly advance our understanding of the function of this classic control circuit. So they combine morphological and electrophysiological data. They built the CNS atlas in Lymnaea, and they have this. I don't know where their atlas is. We'll see if they give a link to it. So let's see if we had any comments in the chat. I think we had 3. Okay. Dick says, is there any C. elegans lineage tree that shows syncytial cells? I'm not sure if they have that specifically. I know that they have the transformation from these developmental cells into muscle cells, but I don't know if they treat them in a way that shows the syncytial aspect of it. Yeah, I don't think that they have that specifically. I think it's just people just have the standard lineage tree worked out. But I don't know. I haven't found a paper on that. Maybe someone has written a paper on it, but I'm not familiar with it. Okay, so so these are these synchrotron X-ray imaging, and so basically this is what they're acquiring. So these are the images here. We have these different ganglia. So in the molluscan brain, you have things organized around ganglia. It's not like the mammalian brain where you have these regions of cortex, and so they're a little bit different. So this is the pedal ganglia, and you can see that they have these clusters here. Hold on. Let me get back into here. Okay. So these are the images here. It's a little unstable when you have it zoomed in like this. You have the pedal ganglion, you have the cerebral ganglion here. This is the 200 micron bar, so you can see it to scale. You have the parietal plus the visceral ganglia here. You have the parietal pleural ganglia, the buccal ganglia, and they have these annotations and color coded so you can see where different substructures are. So you can see there's a lot of substructure in each ganglia, and so the buccal ganglia, the raw image is here on the left, the annotated image is here on the right. Then you have these selected annotated sections that are sort of exploded beneath that. So you can see that there's a lot of structure in these ganglia, and they have these pretty nice images that are, pretty clear, and you can see, if you know what you're looking for, you can see what's going on. You can still see that, like, it's hard to determine the cells. If you zoom in and you do some image processing, you can get good cell counts from that. But you can see where you can have these occluded regions in the brain of the mollusk. So that's that. Then we have this figure here that shows the CNS of the mollusks. This is the molluscan brain here, the CNS. The buccal mass is here. You have the nerves here. You have neurons in these buccal ganglia. So these ganglia are these large masses where they have neurons, and they're these information processing centers. Again, very different from the mammalian brain. And then they connected the buccal mass, which is a muscle, up here. Then you have the synchrotron method. So you have the synchrotron, which is, of course, one of these, you usually find them in association with a particle accelerator. So they're these large structures that generate these electrons. They turn them into X-rays. They hit the sample with X-rays. They put it in a resin medium, and they get the image. So they acquire the image here. Then this is an overview of the CNS reconstruction, where you have these ganglia and you have these neuropill or nerve tracts. I think we talked about neuropill in a previous meeting. Basically, they're these tracts of nerves that connect the ganglia in insect brains and in marine invertebrate brains. So you get this sort of connectivity map here, where you see all these ganglia. This is kind of the internal structure, and then these neuropill. And then you have these recordings from the different, I guess they're different neurons, and they show, the electrical activity here. So what they did was they basically got these high resolution images, and then they confirmed some of their data with these electrophysiological recordings. We have figure 2 here, which is nice because it shows these three-dimensional models of Lymnaea feeding ganglia. So this is showing like this sort of orientation in space. We have going from the left side to the right side. This is the dorsal face, and you have this structure here. You have these different orientations: the posterior face, the ventral face, and the anterior face. So you can see the kind of resolution you're dealing with. You also have the lateral face. And so this is where they have this three-dimensional model with the feeding ganglia. So this is this region of the brain where they have this, it performs this feeding function. And so they've done a lot of analysis on this. This is a reconstruction model. I'm not going to play this, but basically it's a movie that shows you, they rotate this three-dimensional model that they've reconstructed from the image, from the synchrotron image data. Okay. So this is figure 3. This is a class of command-like feeding neurons identified with the SXRT atlas. This is their atlas. So they're able, or actually this is a different atlas, and they're able to analyze their structures here. I'm curious if they have a link to the atlas here that they've put together. This is figure 6, which is this SXRT-based atlas of key cell types in the buccal ganglia. And so they kind of go through these different cell types where they have a location in the feeding ganglia in red, and they have a name for the cell. And so L80 or LB1, which is at that red region at the top left of this image, is the dorsal face motor neuron. They have a location, activity, the action potential looks like. They have these fictive feeding cycles, and then they have this connectivity map here, or they have this connectivity map which shows the excitatory and inhibitory cells. So this is where you have B1, and you have the, it looks like they're all excitatory connections coming in from OC, CGC, and N1M. And then B1, the dorsal face motor neuron, has, I think, an excitatory connection to the salivary gland. So you can see that kind of connection map in place. Okay, so they talk about their map. Our map also provides a detailed view of the neuronal organization of ganglionic regions that have so far remained largely uncharted, thanks to the way that nerve bundles accept the ganglia and thus typically hide some faces from view. We refer to these as ganglionic dark sides, which was mentioned in the abstract. Our atlas allows us to examine the detailed organization of neurons in these regions and thus inform functional targeting, leading to the characterization of a type of feeding motor neuron, B12. Notably, this is an example of a behavior-specific motor neuron in the Lymnaea feeding CPG, or central pattern generator. In this case, recruited into the ingestion cycles, but not the egestion cycles, so one function but not another. The likely behavioral consequence of this differential activation is that swallowing actions are enhanced in ingestion but suppressed in egestion, preventing the animal from ingesting potentially harmful objects. And they talk about this SXRT, which is emerging as a powerful tool for mapping components of mammalian networks, but has not been exploited widely for interrogating the brains of simple model organisms. We anticipate that the approach we outline here will readily generalize to various model systems with comparable brain sizes, so other mollusks, crustacea, annelids, etc. For other animal classes, such as many insects, where neurons are often too small for SXRT to reliably resolve detail, it can provide a rapid means to gain overview maps, for example, of neuropil organization, fiber tracts, soma clusters, and other structural features. So these are all features that you find in these invertebrate brains. And so these are kind of, they're very hard to resolve using conventional microscopy techniques. Since the data sets that it yields are relatively small, especially when compared to their nanoscale maps that emerge from the extraordinary but labor-intensive electron microscopy-based connectomics approaches, the generation of an accurate overview atlas is rapid and easy to interrogate, aiding comparative studies and detailed functional characterization. So this is where we might want to look at inter-individual variability within a species. So we might want to collect multiple connectomes for different organisms and look at the variation there. So we might have a hypothesis about whether something is always generated the same way as it is in C. elegans, and we've confirmed that with data that basically the lineage tree and development lays out more or less the same morphology in every single organism. If they have a defined mutation, usually it has some systematic effect on that phenotype. And using this kind of imaging, you can find out in organisms like marine invertebrates or insects, if that also holds true in those organisms, or if it doesn't, to what extent it doesn't. So, we can share maps across the scientific community. We can use GPU accelerated image sharing platforms, which allow people to manipulate the raw image stacks. So a lot of times we want to share the raw image stacks and not just like summary statistics with our collaborators, so that they can look at different different parts of the image or of the structure and so forth. So this is a bit more powerful, the pipeline that they're developing for this. Let's see if I can find these references up here. They mentioned that SXRT is emerging as a powerful tool for mapping components of mammalian networks, and they point to these references 15, 16, and 68. So 15 is this reference by Bosch et al. Functional and multi-scale 3D structural investigation of brain tissue through correlative in vivo physiology, synchrotron microtomography, and volume electron microscopy. That's in Nature Communications. 16 is sample preparation and warping accuracy for correlative multimodal imaging in the muscle factory bulb using 2-photon synchrotron X-ray and volume electron microscopy. That is by Zhang et al. Frontier Cell and Developmental Biology. So 2 papers where they're doing this synchrotron imaging. And then 68 is here. This is also from Bosch et al. Non-destructive X-ray tomography of the brain tissue ultrastructure. And that's a bioRxiv preprint. So there's, I think, sort of a momentum here for this type of approach. And like I said, again, we've discussed this in previous meetings where, we've talked about doing this in different embryos. And they're not doing this in embryos, they're doing this in brain structures. But you could easily see how this could be modified for looking at embryogenesis. You wouldn't have these functional maps. You might have some electrophysiology, but you would definitely have, I guess you could probably work out lineage trees from these data. So yeah, that's in molluscs. So it's not C. elegans. It's a far different organism, and it has actually quite a bit more complexity than C. elegans. Okay, can someone generate the C. elegans lineage tree syncytial cells? I'm not really sure if they can from the existing data sets that are out there, but I'll look into it. I don't know, maybe. So did we have any questions about the second paper?
Hello, Bradley. Am I audible?
Yeah.
Oh yeah. So yeah, it's Aniketh. So I was going through the graph neural network stuff, and from last year's, which Paki implemented, and I had a question, which was, did she train on just one sample, as in one cell, or was there because I believe she said she did.
Well, she trained on one C. elegans, I think. It's just kind of the C. elegans embryo is, you have this data set where you go from a single cell to, I think she trained it up to 60 or 70 cells. So it was it wasn't the full data set, it wasn't all development, but she did train it upwards to something where you had a pretty robust network. So I don't know.
And
I don't know where her training data is. I'm sure she has it on. I don't remember. I haven't looked through the GitHub repository on that, but we have training data, so it's really kind of training it on these cell centroids and then getting the algorithm to work. But I'd have to get more information from her, I think, on this specific.
Okay.
Yeah.
So she particularly worked on the spatiotemporal aspects of the of creating the network itself, of the graph itself. So I was wondering whether you could suggest some things related to other learnable parameters. So I was thinking whether we could kind of consider gene expressions and cell interactions for training a different network.
Yeah, we could look at. I mean, we've talked about that. The thing about gene expression is, of course, you need to have a data set that gives you good gene expression for development. It's kind of iffy. I mean, there are atlases out there, but it's kind of it's hard to make that mapping. But we could do it. I mean, we could get like some select data and put it in there. Yeah, but I mean that's a different data set, and you need to train it. Or you,
Oh, okay.
yeah, yeah. All right.
So I was like thinking that it was part of the same data set. It was just something like another part or like another parameter that we could like try and model after, like get better results.
Well,
But
yeah,
that's how it.
oftentimes you have to combine data sets, right? You have to like have one data set where you have like the cells, and then another where you have, oftentimes they'll have like gene expression for a single cell or a gene expression for a single region. So you just attach that onto that data set and then use that as like the training data set and the testing data set. You have to make that split and then go for it. You have to make sure you're consistent in how you've integrated those data sets. So that's the only caveat there.
Oh, okay.
Yeah.
Okay.
Okay.
Okay. Yeah.
Yeah.
Thank you.
You're welcome. Okay. Jayadratha said, I am not in the DevoWorm Slack group. Yeah, I think I remember Mehul saying something about this. I can add you. Yeah, I'll add you into the Slack group after the meeting. I'll send you an email. I have administrative permissions for the Slack, so we'll see if I can do it. All right, thanks. All right. Oh yes, this is okay. This is the paper. What is this paper? This is I think this is the paper that. Yeah, this is the Najaro Risi paper. This is the one that, what's her name? Or this is the name that we drew from for the GSOC project last year. So that's a good paper to go through if you're interested. That's the neural developmental programs approach. So building the networks, each cell is a neural developmental program, and you're building these networks that grow. So then the cells are all, I guess, these NDPs, and they're generating this network. So you'll have to go back and read the documentation, not only for last year's project, but back to their documentation for the forked stuff, because this is a fork of that project. Okay. Yeah, thanks. If there's nothing else, thanks for attending. Have a good week.